Purity analysis for downstream laboratory scale purification performed using standard non-disposable equipment. Approximately 10-liter virus harvest from the upstream processing was used. Approximately 44-liter virus harvest from the 50-liter culture was used for further downstream processing with ready-to-process equipment in the scaled-up purification process. Impurities originating from the host cells were removed. During the downstream process, cell debris was removed in the first microfiltration step using ultra prime GF2 micrometer and 0.6 micrometer normal flow filters, followed by host cell DNA removal using a ready to process capture chromatography column. Host cell proteins were removed using a ready to process Capture 700 chromatography were removed. Capture 700 chromatography resin was used in the second chromatography step of the purification process. This chromatography resin has an inert outer layer of cross-linked agarose preventing molecules with a molecular weight larger than approximately 700,000 enter the core of the bead. The bead core contains the multimodal function of the chromatography resin, utilizing both size exclusion and affinity binding, reduces the number of purification steps needed by combining two procedures in one. The properties of octylamine ligand allowing for binding of impurities over a broad pH range, high salt concentrations, and in various buffer compositions enable the use of Capta-Core 700 chromatography resin over a wide variety of conditions without impairment of its function. Hence, a sample from a previous purification step can be loaded directly on the Capta-Core 700 column without the need for pre-adjustment of buffer conditions for our study. These benefits enable decreased number of buffers needed, the possibility to serially connect before the still final step, the viral particles were concentrated and transferred into the final buffer by diafiltration using a ready-to-process hollow fiber cartridge. Finally, the solution was sterilized by filtration using ultra-pure downstream scale purification of influenza virus using single-use Downstream scale-up purification of influenza virus using single-use bioprocessing equipment.